Hi, folks. Dr. Debbie Morris. This lecture is going to be recovering some things that we have touched on in past lectures, but in a little bit more depth. So, we're going to talk about muscle physiology and muscle contraction, and then we're going to talk about a little bit about bone. So, we've touched on muscle contraction in the neurological segment of the course along with cardiology. We touched on bone in endocrinology largely as we've discussed vitamin D and parathyroid hormone and calcitonin. So here we're going to be going into a little bit more depth to round out your knowledge of muscle and bone physiology. And we've got two sets of lecture objectives, which you'll notice to help you with your studying. So, starting with skeletal muscle, and there are a lot of similarities between the different kinds of muscles, and we'll talk about those as we go along. But for purposes of this lecture at the moment, we're going to talk about skeletal muscle. And skeletal muscles are made up of muscle fibers, and the fibers have subunits. So each set of fibers to a point has progressively smaller subunits of fibers, and we'll talk about that structure and why it is effective in producing muscle function, muscle contraction. Outside of any given cell is a plasma membrane called the sarcolemma. So this plasma membrane has a specific name. It's the sarcolemma. Within the sarcolemma, there are fibrils, myofibrils, muscle fibers that are composed of these, largely of these two proteins, actin and myosin. The cells are not individual cells so much as they are a set of fused cells with multiple nuclei. And at the ends of the cells, of the fibers, there are proteinaceous bundles made of collagen that are called tendons. And tendons attach muscle to bone, and that's something that you've learned in anatomy. The cell membranes, or the sarcolemma, like the cell membranes in neural tissue and in the specialized muscle tissue of the cardiac system, these cell membranes maintain a charge and they conduct action potentials. So here is a diagram showing these progressively more detailed subunits. So at the smallest level, we have what we would call a myofibril, which contains myofilaments of actin and myosin, and we'll talk more about those in a minute. So the actin and myosin are the myofilaments. They are bundled together into something called a myofibril. And I want you at the moment to notice these tube-like structures. We'll come back to them in a couple of minutes, okay? These myofibrils are bundled into what we would call a muscle fiber. The muscle fiber being a group of cells with perhaps fused cells with multiple nuclei that is surrounded by a plasma membrane that we would call the sarcolemma. And the nuclei here you can see are eccentric. They're along the edges of this bundle of muscle fibers of myofibrils. So we have myofilaments, myofibrils, we have a muscle fiber surrounded by a sarcolemma, the muscle fiber being this set of cells which are fused together with multiple nuclei. these cells or muscle fibers are bundled together into a structure that we would call a muscle fiber bundle. And nerves and vessels surround the muscle fiber bundle. These bundles are bundled together into what we would call a muscle. So at the ends of the bundles of bundles that we end up calling a muscle, the collagenous ends of those myofibrils and muscle fibers converge into a collagenous fibrous structure called a tendon. That's made up of connective tissue of proteins that are produced by fibroblasts. And the entire muscle is also surrounded by connective tissue made by fibroblasts, which is continuous with the tendon. So this is the structure of skeletal muscle. Actin and myosin are protein polymers, so they are large proteins that are polymerized into fibers, and that attach, the actin attaches to a structure, a proteinaceous structure called the Z-disc. And the actin fibers, which are the thinner fibers, are fixed on the Z-disc. The myosin fibers, which are the thicker fibers, are intercalated with the actin fibers, so they are between the actin fibers, and they have interactions with the actin fibers. You notice that there are charges on the actin fibers that are more positive near the Z-disc and more negative near the termination. The interdigitation of these is going to cause something that we'll see microscopically called bands when we look at muscles in particularly in using electron microscopy, but also light microscopy. You see this structure as bands which stain differently and have different densities. So you can see why that you might, looking at this microscopically, see sort of stripes, and those stripes are called bands. When the signal to contract is received at the neuromuscular junction, and the impulse is conducted through the sarcolemma, which is the plasma membrane, there is the release into the cell fluid, the cytosol, a lot of calcium. And if you remember, calcium is not normally present in the cytosol. It is kept in the extracellular fluid and within the sarcoplasmic reticulum, or in other cells, the endoplasmic reticulum. So normally this cytosol is depleted in calcium, but when calcium comes in and with ATP, we find that the actin and myosin interact in such a way as to contract the muscle. This is an electron micrograph, obviously, of a microfilament, and here you can see the bands. So if you remember, let's go back here, you have the Z-disc, you have actin attached to the Z-disc, you have myosin interdigitated with the actin. Here you can see that you have the Z-discs, okay? Remember that there is actin coming out from each of these Z-discs. There are a lot of mitochondria in this stripe because we need mitochondria to make the ATP that powers the movement that causes contraction. In this I band, we have only actin. So we have the Z-disc in the I band. And then, whoops, these dark stripes are myosin. And the myosin is interdigitated with the actin. The M line contains only myosin. That's why it's the M line. And the unit from one Z disc to the next Z disc is called a sarcomere. So here we have bands. This band is actin. This band is the Z disc. This band is actin only. These bands are actin interdigitated with myosin. The M line is myosin only, and so on. And then in between, lots and lots of mitochondria. Okay. So how exactly do the actin and myosin interact? Well, on the myosin fiber, there are these heads, and these heads reach out. So again, we have the actin filaments and the myosin filament. The actin filaments, as you can see here, are attached to the Z line at this end. The myosin filament will cross between the actin fibers, and the M line will be the portion that's myosin only. On the myosin filament are these little, they're called heads. I'll call them hands. But they are hinged protein structures that form cross bridges to the actin filaments. And when the contraction occurs, the heads or hands or arms use ATP to contract and to bend at the hinge and to move along the actin filament via these cross bridges to cause muscle contraction. Now we're going to come back to something that we've talked about before, the neuromuscular junction. And you remember that nerve endings for skeletal muscle have a junction with the muscle fiber near the end of the nerve. And generally there's one junction per muscle fiber. So any given muscle fiber will be innervated by a single nerve ending. Within the nerve ending are synaptic vesicles that contain ACH, so acetylcholine, because acetylcholine is the neurotransmitter which is important in muscle contraction. The acetylcholine is stored until the action potential reaches the termination at the neuromuscular junction, when voltage-gated calcium channels open, releasing calcium into the extracellular fluid and causing the synaptic vesicles containing acetylcholine to fuse with the plasma membrane and release acetylcholine into the neuromuscular junction. The muscle fiber then has a sarcolemma, which is going to contain acetylcholine receptors. And here we have receptors that you can see on the sarcolemma. So the acetylcholine diffuses into these clefts in the neuromuscular junction. And the next step is that acetylcholine gated receptors cause the release of a great deal of calcium, and we'll come back to that in a minute. this is a picture showing you the anatomy of the neuromuscular junction, where we have the motor end plate, which is the part of the muscle fiber that's innervated by the motor neuron. In the terminal axon, mitochondria is used to synthesize acetylcholine in the cytoplasm, but then it is absorbed into those vesicles and stored in those vesicles. When it is released into the neuromuscular junction in this synaptic space, there's a lot of acetylcholinesterase. So while the acetylcholine binds to receptors in that space, at the same time it is being destroyed so that the release causes a brief response, not a large response. And here again, you see those clefts with the folding of the plasma membrane called the sarcolemma. The vesicles that are formed by the Golgi apparatus are actually transported to the neuromuscular junction. When the action potential arrives at the nerve terminal, calcium channels are open. So suddenly there's a lot more calcium inside, and that calcium increases the rate of fusion of the acetylcholine vesicles with the terminal membrane. I think I had that backwards a couple slides ago. So calcium obviously is not going out of the cell, it is coming into the cell. There are voltage gated channels that are suddenly opened, and calcium surges into the terminal axon, which causes the acetylcholine containing vesicles to fuse and rupture, allowing the acetylcholine to be released into the synaptic space, where some of it binds to receptors and the rest of it is split up by acetylcholinesterase. So it's important, this is a very active process in which acetylcholine vesicles are being continuously produced by the nerve ending. So we now have the release of the acetylcholine into the neuromuscular junction, where there are receptors in these clefts. So these folds of the sarcolemma, within those clefts are acetylcholine receptors. When the acetylcholine binds with those vesicles, they fuse with the membrane. Sorry, here we're still releasing. So the same thing. The nerve impulse releases the vesicles. There's voltage gated calcium released into the brought into the cell, into the axon, causing the fusion of the vesicles with the cell membrane on the terminal axon, causing the release of acetylcholine into the neuromuscular junction, where some of it makes it into these subneural clefts, which are lined with acetylcholine receptors. Now, the acetylcholine receptors also are gated channels, and these are proteins that are lying kind of in a circle in this little tube. And once two acetylcholine molecules attach to a channel, then the channel opens and you have an influx of sodium and the beginning of an action potential, and that action potential spreads, and that spreading of that action potential is going to initiate muscle contraction. And then again, the acetylcholine that has been released is mostly destroyed by acetylcholinesterase, but some of it just diffuses out of the synaptic space. And it's important to prevent the kind of prolonged contraction so that the muscle can recover from its initial action potential. Generally, you can get fatigue at that neuromuscular junction because the impulse is more than it needs to be, and eventually we can deplete the acetylcholine vesicles so that we can no longer pass impulses into the muscle fiber. The resting membrane potential in the skeletal fibers is similar to the resting membrane potential in the large myelinated nerve fibers. So it's a significant negative resting membrane potential of minus 80 to 90 millivolts. When an action potential occurs, it lasts longer in muscle fibers than it does in nerve fibers. So 1 to 5 milliseconds, which is about 5 times as long as the action potential in a large myelinated nerve. And the velocity of the conduction is slower than it is in a large myelinated nerve. So the velocity of conduction of the muscle action potential is 3 to 5 meters per second, and that's about a 13th of the velocity in a large myelinated nerve fiber. Now, back when I showed you the picture of the anatomy of the kind of progressive fibers from the actin and myosin filaments to the myofibrils to the muscle fibers to the bundles to the muscles, I told you to pay attention to these little tubes that were running around the myofibril. So these are called T-tubules, and they're continuous with the sarcolemma. And they are the way in which the action potential gets deep into the muscle so that the action potential can be transmitted efficiently to all of the muscle fibers. So the T-tubules penetrate all the way through the fiber, and the action potential as it spreads along the T-tubules causes the release of calcium ions. So there are voltage-gated calcium channels that are inside the muscle fibers and around the myofibrils. And it's those calcium ions that then cause the interaction between the actin and myosin filaments using ATP that results in muscle contraction. And there's a name for this, which is called excitation contraction coupling. So these tiny T-tubules begin at the cell membrane and are continuous with the cell membrane. Essentially, they are cell membrane, or they're tiny tubes of cell membrane. And here you can see like little pores in the sarcolemma, which are the beginning of these T-tubules, and they penetrate all the way through to the muscle fiber. Now, the muscle fiber, if you remember, is the cell, and the cell has around the myofibrils sarcoplasmic reticulum, and the extracellular fluid is within these T-tubules, and extracellular fluid, if you remember, contains some calcium, as does the sarcoplasmic reticulum. So when the action potential spreads over a fiber, a potential also spreads along the T-tubules. The sarcoplasmic reticulum has these chambers that are called the terminal cisterns, and they are adjacent to the T-tubules. So there's the sarcolemma on the outside of the fiber. There are T-tubules that are extending to each fiber that are adjacent to the cisterns of the sarcoplasmic reticulum. And here again, you can see this picture where we have our filaments of actin and myosin that are bundled together into a fiber, which is essentially a cell, although it is a number of cells. Each of those cells, the bundle of fibers of myosin and actin, is surrounded by a network of sarcoplasmic reticulum with cisterns spaced along. The sarcolemma, or the cell membranes, are continuous with these tubules called T-tubules that are going to enter and contain extracellular fluid and end up surrounding the cisterns of the each individual of the sarcoplasmic reticulum of each individual muscle fiber. And then again, there are lots of mitochondria in here surrounding the cells to produce the ATP that's going to be important in muscle contraction. And there are nuclei that are eccentric. So this is not a single cell. It is a merging of cells with this structure of myosin and actin fibers that are arranged in the structure we call this is our sarcomere from C disc to Z disc, and that those filaments are bundled together and surrounded by nuclei, mitochondria, and a sarcolemma. So the excitation contraction coupling occurs in the cell, in the cytosol, which in a muscle is called the sarcoplasm. So cytoplasm, sarcoplasm, and it's mediated by calcium ions. So first, if you remember, we have an action potential generated at the neuromuscular junction where acetylcholine merges with acetylcholine receptors, which opens acetylcholine-gated sodium channels, which start an action potential down the sarcoplasmic reticulum. That action potential travels down the T-tubule and activates receptors in the T-tubule that are voltage gated. They're called DHP receptors, and they cause the release of calcium by opening calcium channels in this sarcoplasmic membrane in the T-tubules. And so now calcium ions are going to flow in to the cytosol of the muscle fiber, and calcium ions are going to flow out of the sarcoplasmic reticulum. And they, so the calcium ions are now flowing from channels in the sarcoplasmic reticulum into the cytosol and cause the actin and myosin. They bind to the actin and myosin on a protein that's called the troponin C subunit, triggering the, along with ATP, the bending, the hinging of these heads and moving the actin towards the M line. And then there are pumps in the sarcoplasmic reticulum walls that are constantly pumping calcium back out of the cytosol or sarcoplasm and into the sarcoplasmic reticulum. I don't care that you know a great deal about this. You don't have to certainly know the details, but you do have to understand that a couple of things. One, the importance of the initial of the ACH, the acetylcholine receptors, which are a chemically gated receptor that cause an action potential in the in the muscle cell. That action potential is transmitted through T-tubules to the interior of the muscle fiber, where there are voltage gated channels that open, that cause release of calcium, largely from the sarcoplasmic reticulum, but also from this little bit of extracellular fluid that is in the T tubule. That calcium interacts with the myosin and actin, and causes the use of ATP to contract to move the myosin fiber, to pull the actin fibers closer together. Okay, so here we have our cell body with dendrites that are receiving messages, with an axon which is sending messages, with terminal axons that are attached at the neuromuscular junction that are releasing synaptic vesicles containing acetylcholine when the action potential reaches the terminal axon. In this case, it is calcium influx because there are voltage gated channels that cause the merging of these vesicles with the plasma membrane to release acetylcholine into the neuromuscular junction, where there are, within these clefts, acetylcholine receptors, so chemically gated receptors, that then cause the opening of, that cause an influx of sodium, which results in transmission of the action potential along the sarcolemma, or the plasma membrane, where it is transmitted into T tubules. The T tubules contain voltage gated channels that are going to that are in contact with the sarcoplasmic reticulum and are going to cause the release of a lot of calcium ions, and from the T tubules, and those calcium ions are going to cause these bridges between the myosin fiber and actin fiber to result in muscle contraction. Now we're going to talk a little bit about bones. So that's enough about muscles. That's how muscles work. Bones are extremely important for obviously a lot of reasons. One reason why bones are really important is to hold us up and to hold us together, clearly. But another is that most of the body's calcium is stored in bone. In order for the organism to maintain homeostasis, calcium is very tightly regulated. So it's regulated by things like parathyroid hormone and vitamin D and calcitonin. And because of that regulation and the importance of bone to store calcium, PTH, vitamin D, and calcitonin are all very involved in bone physiology, and we've talked a little bit about that. We'll come back and talk more about that. Now, phosphate is also in large measure stored in bone because hydroxyapatite is a calcium phosphate. There's a lot of phosphate also in cells, but 85% of the phosphate is in the bone. And it is not as tightly regulated as calcium, but its regulation is tied to calcium regulation. There are essentially three kinds of cells important to bone. One of them is a cell that's called an osteoblast. And as blast cells are cells that make things, they are reproducing cells. So osteoblasts are constantly depositing bone. There are cells that are called osteoclasts, and osteoclasts are reabsorbing bone. So there's a non-stop balance between the deposition of bone and the reabsorption of bone. And sometimes the osteoblasts are more active, and sometimes the osteoclasts are more active. As the osteoblasts deposit bone, they can get trapped within the matrix of the bone, in which case they are renamed osteocytes. So most long bones have a very similar structure. There are long bones, there are short bones, there are flat bones, and there are irregularly shaped. But within long bones, we have a constant structure. Inside of the long bone are two kinds of marrow. There's red marrow, and red marrow is largely within these cavities in spongy bone. And there is yellow marrow, which is largely contained within the central cavity of the long bones. There are membranes. There's a membrane on the outside of the bone that's called the periosteum, and there's a membrane that lines the inside of the bone and of each little bony cavity, and that is called the endosteum. There is a kind of bone that is trabecular or spongy, which we call spongy bone or cancellous bone or trabecular bone, and which contains these little cavities that contain red marrow and are continuous with blood vessels. And there is bone that is much more dense, that is called compact bone or cortical bone, which is largely the structural component of the bone around the outside of the bone and containing the spongy bone and the cavity in the center of the long bones. Here we see a picture where we're sort of expanding the structure of the bone so that you can see our bone with its compact bone on the outside and surrounding some spongy bone on the inside. The periosteum on the outside and the endosteum on the inside of each of these pieces of bone. There are vessels that enter the bone. And over here we can see these blood vessels. There are some that perforate from the outside, and then we branch into these structures that are called Haversian canals, and within the Haversian canal is a artery and a vein, or an arteriole and a venule, I guess an artery and a vein, and there are little canals called canaliculi and layers of bone that is being formed by the osteocyte, and this creates a unit called the osteon. Let's look over here and see what else we can see. There are also nerves within the Haversian canal. And you can see how the bone is being made in layers and how osteoblasts end up being trapped within the bone that they're making and becoming osteocytes. So the bone is not only minerals. It's about 70% minerals, hydroxyapatite, and it's about 30% matrix. You need proteins, basically connective tissue, to hold this stuff all together. It's the glue. So that matrix, which makes up about 30% of the bone, is largely formed of collagen fibers that extend along the lines of force of the bone. In addition to the collagen fibers, we have something called ground substance, which is largely proteoglycans, um, gelled proteoglycans in water and extracellular fluid. And then in addition to the calcium phosphate, the hydroxyapatite, we have other ions. There's some magnesium, obviously sodium and potassium, because we always have sodium and potassium, and some carbonate, which is also um, contains calcium. The collagen fibers are a big contributor to the strength of bone, both to its tensile stretching strength and its compressional strength. And the collagen fiber binds to the hydroxyapatite fibers. So it holds this all together. It's the glue that holds the crystals of mineral together, so that the collagen fibers and hydroxyapatite are bound. They can't slip out of place. And they overlap a little, and it's the collagen fibers that provide the tensile strength, the bending, stretching strength, and the calcium salts that contribute to the compressional strength so that you can't compress a bone easily. Hydroxyapatite only precipitates in bone tissue. There are inhibitors in all other tissues to prevent precipitation of hydroxyapatite. If otherwise, we turn into great big rocks or boneheads. We don't need that. So how does that happen? Well, osteoblasts they're making collagen molecules, right? What do cells do that are anabolic cells, that are creating cells? They make proteins. And in the case of osteoblasts, they're making and secreting collagen and ground substance, which are proteoglycans. And the collagen monomers polymerize to form collagen fibers. And that collagen only tissue, or collagen and proteoglycan tissue, is called osteoid. Now, some of the osteoblasts get trapped within the osteoid and they become dormant. They can't keep making bone once they are trapped, so then they're called osteocytes. But fairly quickly, calcium salts precipitate on the collagen fibers and eventually become hydroxyapatite crystals that are kind of bonded to the collagen fibers, creating this extremely strong structure. So bone is constantly being deposited by osteoblasts. That's what osteoblasts do. Once the osteoblasts become inactive, they are osteocytes trapped within the osteoid and trapped within the bone. On the other hand, there are these other cells that are called osteoclasts, which absorb bone when they're active. Osteoblasts occur on the outer surface of the bone under the periosteum and also in all the bony cavities. And there's always some osteoblastic activity in all living bones. Bone is constantly being remodeled. In the case of long bones, certainly during childhood, there's constant remodeling because there's the necessity for the bone to grow. And so there is a physis, or a growth plate, and next to the physis is the epiphysis, which is the growing tissue that's enlarging as this cartilage grows and is replaced by bone. And ultimately, as this cartilage grows and is replaced by bone, the shaft of the bone will elongate. And this diaphysis, so diaphysis, epiphysis, and physis, and again epiphysis, is where bone lengthening is taking place, and there's a lot of very active bone formation. As bone is growing, and here you see sort of this original bone, which is now grown to this, you have to also remodel the bone and have osteoclasts, which are resorbing and removing old bone to maintain optimum strength as the bone lengthens. So the osteoclasts are phagocytic. They're cells that eat, and they have multiple nuclei. They are formed in the bone marrow, and they're active in an adult about 1% of the surfaces of the bone. Osteoclasts, like many phagocytes, have projections, and they send out these projections that are sort of like villi that form a border next to bone and secrete enzymes that do two things. One of the things that they do, the proteolytic enzymes, digest, break down the collagen and the proteoglycans that form the organic matrix of the bone, while acids cause dissolution, dissolving of the bone salts. So small particles are phagocytosed, and ultimately the products of this, the amino acids and the minerals, are released into the blood. And here we have a picture of sort of bone remodeling involving osteoblasts, osteoclasts, and osteocytes. So here's a bone in its resting state. It has cells that line it. It has osteons containing osteocytes. When an osteoclast becomes open, it sends out its projections and begins to reabsorb bone, to break down the matrix and break down the mineral and phagocytose. As this begins to reverse, mononuclear cells in the bone marrow turn into osteoblasts, and those osteoblasts begin forming new bone. So they're going to be making lots of matrix, so collagen monomers, which are excreted, proteoglycans, which form gels with the extracellular fluid. The monomers polymerize into collagen fibers. Those fibers make osteoid, which is then mineralized as hydroxyapatite precipitates, and we end up with new bone in its resting state. So this happens whenever a bone is cracked or broken. It happens a lot during periods of rapid growth. It happens when new stressors are added to bone, such that the tension lines are changed. So under normal circumstances, the deposition of new bone and the absorption of old bone are in equilibrium, and we have a constant bone mass. The osteoclasts come in masses or to areas that need breakdown. And once the breakdown is complete, the osteoblasts enter and lay down new bone in those lamellae and concentric circles in the cavity until it's filled. And when the blood vessels supplying the area are completely surrounded by bone, that's an osteon with a lamellar cavity. It's these concentric circles, and the new bone is now made and stable. So bone has to continually remodel to adjust its strength to the degree of stress that's being acted on it. And in addition, the shape of the bone can change and be rearranged according to the stress patterns. So say somebody starts limping and they're less using one leg than the other leg, say because of a foot injury. The leg that is being used more, that bone is going to have to remodel because of different stress patterns of stress that are being applied to it as the person limps. Old bones become brittle and weak and need new organic matrix to replace degenerated matrix. In healthy people, the control, well, in all people, the control of the rate of bone being deposited is by the amount of stress and the lines of stress that are being applied to the bone. So the compressional load is going to determine how much bone is deposited, and people who are athletes tend to deposit more bone. People who become sedentary and are not bearing weight don't deposit bone and have net bone loss. They have decalcification. So when you put somebody to bed, they immediately begin losing bone. When you get them back up and are putting weight on bone, it's going to start redepositing. This is why early weight bearing after fractures is usually encouraged. There are situations in which that's not helpful, but orthopedists work hard to make sure that they immobilize a fracture but allow weight bearing in order to get early deposition of new bone. The stress and tension on a bone can cause bone remodeling. So when somebody has a fracture that heals at an angle, over time that angle often corrects, particularly in young people. This is less so in older people, more so in younger people, but the lines of stress cause bone remodeling, both breakdown and deposition, that ends up in correcting an angulated fracture. Whenever a bone is broken or cracked or fractured, all the osteoblasts in the area get activated, and new osteoblasts are formed from the progenitor cells in the bone marrow. And immediately you start getting deposition of osteoid and then mineralization. So you have a bulge of tissue that's the osteoblasts and the osteoid, the new organic bone matrix, and then you get deposition of calcium salts, and this bridges the two broken ends and is called a callus. So here we have a fracture site, we have some bleeding and a hematoma, and you end up with increased blood flow and increased permeability of the vessels. You have fibroblasts that are creating a mesh of fibrin, lots of different mesenchymal stem cells that are going to differentiate into osteoblasts, revascularization, so you're going to have vessel growth factors that are going to cause blood vessels to grow into the area. Then you're going to have osteoid formation, which would be the organic matrix and is called a soft callus for 3 to 4 weeks. And then mineralization takes 3 to 4 months to get full mineralization of the osteoid. So you have a new mineralized bone. And then over time, and this can take months to years, the deformity, the hard callus, is reabsorbed by osteoclasts, and new lamellar bone is deposited by osteoblasts in accordance with the lines of stress on a weight-bearing bone. There are a few problems that can inhibit proper healing of a bone. So, displacement, when the bone ends aren't adjacent to each other. This process is going to be much harder. You can imagine if this bone end is over here and this one is over here, you're not going to get this bridging and vascular entry and formation of new bone. Instability. So maybe those ends are next to each other, but the break is through and through, and every time force is applied, there's some movement. That's going to prevent proper healing. So displacement would be why a bone would be set or reduced. A fracture is called reduced when the bone ends are brought adjacent to each other. Instability is treated by stabilizing, which can be either externally with something like a cast or internally with something like a pin or plates and screws. When the blood supply is poor, fracture healing is slowed, and this is often a problem in people with vascular disease, including small vessel disease from things like diabetes and larger vessel disease from peripheral arterial disease. Now, vitamin D is super important. When vitamin D is an fat-soluble vitamin, which in excess amounts actually has toxicity. In excessive amounts, people taking way too much vitamin D can develop hypercalcemia, hypertension, constipation as a result of the hypercalcemia, and abnormal calcifications as a result of the hypercalcemia. When there is too little vitamin D, then PTH doesn't work very well. And so if parathyroid hormone is unable to play its part in bone, the bone will be absorbed or reabsorbed in high amounts, ending up in osteopenia and osteoporosis. So in proper amounts, vitamin D promotes the calcification of bone and is really important in the absorption of calcium and phosphate from the intestines and the mineralization of bone. Parathyroid hormone, as you remember, has the effect of causing absorption, reabsorption of calcium and phosphate from the bone. It has two effects, a rapid and a slower effect on the bone. The rapid phase happens very quickly within minutes and increases over hours and activates bone cells that already exist. The slower phase takes days to weeks and results from osteoclasts proliferating. So more of these bone breakdown cells are produced, causing more reabsorption of bone by the osteoclast. So during the rapid phase, PTH causes bone salts to be removed from bone matrix near the osteocytes within the bone and near the osteoblasts along the surface of the bone. The osteoblast and osteocytes have a system of interconnected cells with membrane pumps that pump calcium out of the bone fluid into the ECF when creating a concentration. And when the amount of calcium in that bone fluid is too low, bone is broken down, calcium and phosphate are absorbed from the bone, and this is called osteolysis. When that pump is inactivated, calcium concentrations go up and calcium phosphate salts are redeposited in the osteoid in the matrix. So what does PTH do? Okay, in the slow phase, as the osteoclasts are activated, a signal is sent causing from the osteoblasts and osteocytes, causing bone reabsorption. So we both activate the osteoclasts that are already there and we make more osteoclasts. And when PTH, parathyroid hormone, is secreted in high quantities over a long period of time, we'll get bone reabsorption, we'll get cavities in the bone, ultimately we'll get osteoporosis and fractures of the bones. And then calcitonin. Do you remember what calcitonin is and where it's made? So calcitonin is a hormone that has the opposite effects of PTH and is actually made in the thyroid by specific cells within the thyroid that make up a small minority of thyroid cells. It has, when calcium concentrations in the extracellular fluid in the blood go up, calcitonin secretion is stimulated. If you remember, PTH is stimulated by decrease in calcium concentrations. So the combination of calcitonin and PTH provides this feedback mechanism that controls plasma calcium concentrations within the necessary tight range to maintain normal functioning, not just in the skeletal system, but in all of the systems in which calcium is important, particularly neural and neuromuscular. So when calcitonin is released, it causes the osteoclasts to slow down and a decrease in the formation of new osteoclasts, so that we are no longer releasing calcium into the extracellular fluid and blood by breakdown of bone. So it slows bone breakdown.